This meeting is being recorded.
Great. So welcome to From the Ground Up, everything you need to know about seed libraries. This session is about advanced seed saving, brassicas, and we're scheduled to go until 10:30 a.m. Before I get into it, I'm just going to acknowledge that I'm calling in from the traditional and unceded territories of the Kikite and Coquitlam First Nations, but I recognize we're all on different territories, and I would ask you to take a second to just appreciate the land you're on and acknowledge who it belongs to. So humans and Indigenous people have had a close relationship to seed throughout time. Their seeds are part of our collective inheritance, often seen more as a relative rather than a commodity. And it's part of our duty to care and nurture the seeds as they done for us. So yeah, thank you for listening to that seed acknowledgement. So next I'll pass it over to David, but just to give you a bit of background about him. So David started his farming career in Vancouver, working with the Environmental Youth Alliance, running gardening programs for youth out of the Cottonwood and Strathcona community gardens. He had the opportunity to experiment with seed growing, breeding, low-till and annual production, intercropping, and companion planting while working with Glorious Organics Co-op in Aldergrove, BC. He has taught workshops in gardening, composting, permaculture, and seed saving to adults and children all over the Lower Mainland. And now he works as our BC Seed Security Manager at Farm Folk City Folk. So I'll pass it over to David to learn a bit more about brassicas.
Well, thank you, everybody. I was wondering who would show up at 9 o'clock in the morning on a Saturday to listen to a workshop about brassica seed saving. So thanks for being here. Yeah, so I'm just gonna, I guess we can sort of get started and dig in. I think Lisa is gonna share a presentation for me, and we will go from there. Do you want to go one slide back? Awesome. Okay. So yeah, just an introduction to this workshop. Give a little bit of background about my learning over the years. I did go to university for biology, but I actually never took a botany course. I did most of this learning while farming for about 20 years after, and doing a lot of seed saving. And through this presentation, I will introduce you to some of my best teachers. Some of them are pictured here on this page.
The votes in the lower house passed with a big...
And I will introduce you to others as we go through. If people can just maybe mute themselves as they come into the meeting, that would be great. And yeah, so I'm just going to go through this. You can skip to the next slide. So for starters, we're actually going to just launch, we're going to launch a quick poll just to see who's in the room. I'm kind of curious what you're interested in learning. And this slide, I'm just curious, do you consider yourself a seed saver? Are you a seed breeder? And what's the difference? And I think what I'd propose through this presentation is I hope that we can all think of ourselves as seed breeders. It doesn't mean you have to work in a lab. Seed breeding is something that's been done for thousands of years. And it's done in farmers' fields, and it's done through careful observation and in a lot of ways just learning from the plants. I encourage you to delve into some kind of breeding project, and you learn a lot through that process. I would also say that if you're a seed saver and you want to do a really good job of saving seeds, it's really beneficial to understand how to do seed breeding as well. Because in seed breeding, we're often crossing different varieties together. In seed saving, you want to make sure you don't cross specific different varieties together. So in order to do a good job of that, you need to understand how those varieties cross. And then even in the seed selection process, even if you're just saving an old variety and you want it to be improved over the years, it's really beneficial to actually understand how you're improving those crops. And that takes careful observation each year and deciding which individuals you're going to save for seed and which you're going to rogue out and not let go to seed. So that this will help us create new varieties, but it'll also help us improve existing varieties. So I'm not sure if you can launch the poll.
I hit launch,
Oh,
so
I think I can, so I'm gonna launch it.
I can see results.
oh great.
Okay,
So
cool.
far, we've got 12 out of 18 people voted. I mean, I could end poll and it might show it up to people, but we could still let the last few people. But it seems like, have you saved Brassica seed before? We've got about a third saying no and 2/3 saying yes. A lot of people have saved kale, broccoli, radish, arugula, mustard. So far, no one with turnip, cabbage, cauliflower, or rutabaga. And then 92% of people identify as a beginner who wants to learn more,
Okay.
and 8% as an advanced, and 33, so a third of people are interested in preserving old heirloom varieties, and 42 are interested to learning new breeding varieties.
Great. That's a great spread. I guess you cover the gamut. And again, if you're interested in breeding new varieties or maintaining heirlooms, I think it's important to think of ourselves as breeders because we're trying to improve varieties over the years. So you can go to the next slide. So just some basics. This is just like a general overall about the Brassicaceae family. So this applies to all of these crops, kale and broccoli and radish. And so seed saving of these crops, it's in our coastal regions, it's a really good area to save seed. The seeds, they mature well in the cold season, and then they need a dry spell to mature up. They usually mature up in mid to late summer for a summer harvest, which is great for us here because that is during the dry season. There are a lot of diseases, the Brassica diseases, and the next couple slides I'm going to talk about some of the diseases. I wouldn't get too worried about it. However, there are some just south of the border, and I imagine as the climate changes and things warm up, we're going to see them shifting north. Seeds are really easy to save of these crops. They can be threshed easily on a tarp. The cleaning is usually done by using screens and winnowing, and they separate from their husks quite easily. So you don't need any advanced fancy winnowing methods. Box fan and some Rubbermaid totes work really well. Birds can certainly be a pest in some areas as the seeds ripen. We often get them wanting to eat up our crop at the seed farm, and in some years we've had to cover them so that we don't lose all our seeds. Often we stake the plants. You don't have to, but if you're really trying to maximize the amount of good quality seed, you want to keep them off of the ground, keep good airflow going. The seeds generally have a really good shelf length. So these aren't seeds you have to save every 2 years. If you save a broccoli seed, you're good for probably 5 to 10 years. Of course, germination is going to go down as you get closer to the 10 years, depending how it's stored, but still pretty good. Seeds germinate really quickly. These are often direct seeded crops that work really well in our climate. And the plants produce abundant volumes of seed. So in terms of seed saving, it's a really good producer. And that we have both annual and perennial, depending on the type of crop. So you want to just go to the next slide and maybe, yeah. Okay, so these are just, this is just a list of some of the diseases. I wouldn't get too concerned about them in all my years of saving seed. I think there was one year that I saw a crop of seed that I actually decided not to save from because it did look like it was covered with a fair amount of a disease that I didn't go and identify it positively, but it made me a bit nervous. If there's always, when you save seeds, there's a little bit of fungus, a little bit of mold, a fair amount of aphids on our crop, usually by the end of the time we're harvesting them. Those are all okay. They'll come out in the seed cleaning process, and then you can it's fine. The main disease that I've heard about of some concern is Alternaria. This is a disease that's actually monitored just south of us down in Oregon and Washington. There's some regions that if you produce seed down there, you actually have to get your seeds tested in order to sell them. And if you just click to the next slide, this is a picture of what Alternaria looks like. You probably, if you look at this and you go, oh my goodness, I've seen this all over my brassicas, it also looks like other diseases as well. So again, it's really important to really positively identify. However, just like saving anything, if I was going to harvest a bunch of tomatoes and there's a lot of disease, it's the tomato itself isn't going to store well. It's probably not going to taste as good. And similarly, if you have a seed crop that has a whole lot of disease over it, it is definitely going to affect the seed no matter what the disease is. So preventative measures for these things, I think, are the most important. We're talking about working in organic systems, and there's no organic control for these. This disease is seed-borne, which is why you just want to make sure you don't have it in your crop. And yeah, there's no seed treatment for it. So allowing lots of airspace between your plants and trying not to overhead water during the warm season are probably two of the best ways to help mitigate that. Okay, so we can go on to the next slide. So this is just a table of some of the family of crops that we're going to be talking about a little bit today. I'm happy to jump into any of these. Through this presentation, some of my examples focus a bit more on the Brassicaceae species. But one of the things I wanted to point out from this chart, so this chart is, this is information in this chart compiled from the book called The Seed Garden that was put out by the Seed Savers Exchange, as well as some information from Mary Alice from Full Circle Seeds. So I've added some info in there about some of the specific varieties, because a lot of the time when we grow varieties, especially of the like salad green, Rapa and Junkiae species, they can look similar and it's hard to know exactly which one fits into which category. And so if you're saving seeds of both, it's important to know which varieties fit into those species so you don't have cross-contamination, or so you do have crossing depending on what you want. The other thing I wanted to point out is like brassicas are predominantly outbreeding. A lot of them are self-incompatible. So you'll see like Brassica oleracea species are self-incompatible. What that means is that an individual, although they have perfect flowers, there's a chemical incompatibility within its genes that doesn't allow it to pollinate itself. So one individual won't pollinate itself, except for a couple of the species. And again, there's always exceptions to these rules. So when I say that, maybe it's at a 99% rate. For the Junkiae and the Napa species, however, those plants do have the ability to be self-fertile. Took me a while to realize this because I would always teach people that, kale, for example, was very outcrossing, and I'd come upon people at workshops that said, but I've been saving, red Russian kale seed for years, and I just have one plant in my garden and it seems fine. And it does seem like that variety does fine without a very big population. You can also get away with small populations for a certain period of time. But when we're talking about improving these seeds over years, it's good to be choosing from a wide population. Because these seeds are mostly outbreeding, we want as many as we can to select from, and we are doing what we'd call population breeding, which means kind of looking at the whole population. It's not that one individual becomes really interesting, like in a tomato, and you can isolate that and then grow it as a self for years after. Okay, so next slide. So the triangle of U is a theory that was proposed back in the 1935 by Wu Jiang Chang. He was a Korean Japanese botanist. This is a theory about sort of the history of how these crops came to us, why we have how they segregated, how they were created. The earliest records of these species being related to modern day cultivars goes back like 3,500 years. So when we talk about crossing these varieties or making new varieties even within one species, this is really slow work. I'm not saying that a new variety will take 3,500 years to produce, but it'll take a decade or two for sure. So you're into a long project if you create a new variety. And I would also argue you'll have a really good teacher over those years because you learn a lot of stuff from doing that. So the next slide. This just shows the potential crossability of these species. Now, this doesn't mean that when you plant them in your field, you're going to have a strong chance of these things crossing. When we talk potential crossability, we're talking about using techniques that probably none of us use in a lab. And it's more about testing the relationship between these individuals or these different species and how they came to be. However, it is possible always or sometimes for cross-species breeding to happen. And again, it's very rare, but obviously over the last 3,500 years, it has happened a bit. It doesn't happen nearly at the rate that today people are crossing species with one another through like more genetic modified techniques. So I'm just going to talk a little bit. This is just in reference to, so when we do think about maybe breeding a new variety, we want to be sort of aware of the parents that we choose. And one of the reasons is because a lot of Brassica species, modern day cultivars, especially hybrids. So this has more to do if you're breeding and using some of the hybrid Brassicas. It's hard to find out, but you need to try to find out if they were produced through this process called cytoplasmic male sterility. The purpose of this is when you create a hybrid, you have two inbred parents. And when you cross those parents, it's really important to know which ones produced the pollen and which ones produced the seed, because across one way isn't going to give you the same result as across the other way. So in order to do this effectively, you could imagine if I'm a producer of hybrid seeds and I have two different lines, breeding lines that I want to cross, and I want to make sure they only cross one way, I'm going to go out to the field and hand pollinate a whole lot of plants. And that is incredibly labor intensive. And there's no way that it's going to pay off. So there's different techniques over the years that people have used to do this crossing that's less labor intensive. Sometimes it's spraying one of the parents with a chemical that will stop the pollen from being able to be transferred so that you know that the pollen transfer just goes one way. But the other way that this has been done in a lot of the Brassica species is using this method. So again, this isn't something that you're going to do at home. You need a lab and you need access to certain chemicals and probably radiation to make this happen. I don't know how to kill the nucleus of a cell and maintain the mitochondria personally. So this isn't something I'm going to do. However, if I'm working with a variety that's being bred this way, I do need to know that. And the reason being that cytoplasmic male sterility will get passed down through the next generations. So you are going to have a whole lot of individuals in that population that are male sterile, so they don't produce pollen. This technique right now is actually quite controversial because currently certified organic bodies, some of them especially in Europe, consider this method of seed production as genetically modified, and therefore these varieties are not allowed within the organic regime. In the United States, that's not the case, and Canada is in the process of debating this currently. And the big advance that happened through this process is when you go to the grocery store and you see broccoli in the store today, as early as back in the 1960s, I don't think you would have even seen broccoli heads the size and the density that they are today, because it's been this technique that's been able to sort of make them that uniform, and sort of the new sort of dense heads, the single cut large broccolis that we see today. So the ones that weren't developed with this technique are more the old heirloom types that are more sprouting broccolis of the past. And personally, I find those ones tastier, and I prefer to harvest broccoli over a long period of time in my garden rather than have all of it mature at once. So the old heirlooms work really well for me. However, the focus on breeding broccoli has very few people have been working on open pollinated broccoli varieties because a lot of the farmers and growers are using hybrids, and so there just hasn't been the work put into maintaining those OPs. So I would argue, I think we can come up with some really good open pollinated broccolis if more of us start playing with them and working on them. So the next slide. So I'm just going to start getting into some of the different species. So this is obviously, this is the biggest category. The Brassica family is one of the biggest families of cultivated plants that we eat today. So in this picture, it's important to keep in mind, all of these will cross with one another. So if you have in your field a broccoli and a cauliflower flowering at the same time, those will cross, they'll make something new. Same with any of these things flowering. The kale here, keep in mind, red Russian kale is in a different category. This is referring to the Oleracea type kale, which is like the lacinato and the European type kales. The harvest window for these, like the others, is during the cold season. Most of these crops are grown as biennials, except for broccoli. So in order to grow broccoli, it's hard for people in British Columbia who live in a lot of the hotter climates, like the interior, to grow broccoli seed very well. It's a bit iffy because by the time it's making seed, it's so hot and dry, and the seed formation really doesn't do well in that climate. So it's really great on the coast here if you can get your broccoli in really early so that seeds are maturing in the cool season. That doesn't mean you can't grow them in other areas, and I'd encourage people to do that. It's through selection, not just of the characteristics for eating, but through characteristics like seed set and selection for plants that make good seed in your location. You can acclimatize these plants to your location over time for sure. So for me, some of the biggest challenges has been storing some of these crops over winter because you do have to grow them as a biennial. So for example, cabbage, you need to save the whole plant, and that requires digging it out of the ground. If you're in a region where you know it's not going to overwinter, if you can overwinter your cabbage in the field, then you're laughing. If you're growing something like a Brussels sprout in your area, most, depending on where you are in the country, Brussels sprouts are extremely hardy and they should be able to get through the winter no problem. So that's one you wouldn't have to pull out of the field and try to store inside. But my challenge in the past has been when I want to save a crop of cabbage, for example, I want to save from, if you refer back to that early chart, I want to save from a population of probably 50 to 80 plants. That's a lot of space. Most people's refrigerators are not going to hold 80 cabbage plants. If you have a walk-in cooler, that's even a lot of room. So this is some of the challenges of growing these crops. What we've tried this year, actually, instead of bringing them into the cooler, is we dug a trench in one of our greenhouses and healed them in. And we're hoping that with the plastic protection and maybe a little protection over them, they'll do fine over the winter. And in our climate here, they mostly do, but you'll have to determine that for yourself. Yeah, okay, so that's some of the Oleracea species, and we can, I'm going to try to get through this slideshow and then we can get into some questions and answers after. And feel free, if you wanted to pop any questions in the chat, I'm happy to be interrupted throughout this presentation. Okay, so yeah, this next slide, this is the mustard species, so the Brassica juncae. There are other mustards. If you grow salad greens, you're probably familiar with Mizuna and Tatsois. Those are in the, I believe, in the Rapa species or the Napa species, and they're a lot more mild. These are typically your spicier mustards, although even through here, some of them are a lot spicier than others. I find golden frills and ruby streaks is often quite mild compared to like green wave and wasabina. Again, all of these crops will cross with one another. It looks like there's a question in the chat. Can you harvest the cabbage head and hope for the plant to flower, or is the head the part that will flower? That's a really interesting question. We're experimenting with that this year. There was a farmer directed study that was done out of the EFAO, the Environmental Farmers Association of Ontario. They did a study where they actually trialed this. So they harvested the cabbage heads and kept the stalk and root. And then they, on one of the populations, the other population, they left the head in. So if you're going to get your cabbage to produce seed after you harvest the head, you do have to cut it high enough on the plant so that there's still growing tips. But what they found was the seeds that were produced from the cabbage that didn't have the heads cut were typically like larger seeds and maybe slightly better quality. They might have had a higher yield, but the seeds from the cabbages that had their heads cut also produce seed, and they again, genetically they're the same. The seeds might have been a little bit smaller, but when they did sort of a cost analysis, if you're a producer that grows both cabbage and cabbage seed, they found that you would probably make more income by selling the cabbage and producing the seed rather than keeping the cabbage on. So it really depends on what you're doing. If you're really working on producing lots of really high quality seed, then it's fine. Then you probably want to keep the cabbage heads on. If you're maybe in a breeding stage, in some ways, it's actually really useful to identify the earliest and nicest heading cabbages by harvesting them. And then you know that those were the best ones and you actually save seeds from those. So genetically, you'd be selecting the nicest heading up cabbage first. So thanks for that question. A root cellar is the question, would a root cellar work for storage? Yeah, I think a root cellar would work fine. The thing I find often gets me in storage is if you don't have storage, the fluctuating temperatures can sometimes affect the crop, and then mold and disease on the leaves. So you probably want to have as few leaves on the plant as you need to keep it through the winter so that there's just less stuff to rot on the plant. I've spoken to one grower who actually in his cooler uses an air purifier. I haven't heard anybody else doing this, and he shared with me, he's like, I haven't verified that it's useful, but I just figure any amount of like pulling out different mold spores and bacteria out of the air is probably going to be helpful. So that might be something to try. So getting back to these mustards crops, these flowers, again, in this species, the juncae, some of them are self-fertile, which means that selfing is possible, and so you might need a lower population of these than other species. And it also means you might, especially in the breeding stage, you might be able to use a much smaller population. And we'll maybe see an example of that a bit later in the slide presentation. So lots of possibility for making new varieties within this. A lot of these sort of frilly, lacy mustards I've sort of seen come available in the last couple decades. So it's not, it's probably been around for a while, but not in the popular seed world. Okay, so the next slide, just looking at some of the different types of radishes. So again, consider the harvest window for radishes. The challenge I've had with this, so one of my teachers has been, their name is Ostergrass radish. That's one that I've saved a couple times. Some of the challenges of saving radishes is they have differing vernalization requirements. So some radishes are quite strongly biennial, and if you plant them early in the year, it's going to take them quite a long time to bolt and to make seed. And some of these varieties won't even do it very well. The red meat or the watermelon radishes and black radishes, those tend to be like really biennial type radishes. They're good for overwintering. They store really well, but it's tricky to plant them early in the year and then get a crop out of them because I believe they need a bit longer vernalization. So what that means is in order to trigger them from their sort of vegetative stage to a flowering and seed production stage, they have to go through a period of cold. And some of the periods of cold for some of the radishes are quite short, like 30 days. So if you pull them out of the ground, you stick them in a cooler for 30 days, and then you replant them, it'll be triggered, the second stage of their growth will be triggered, they'll make seed. And some need a bit of a longer time. Some radishes, I'm sure we've all found radishes that bolt in their first year. So there's obviously radishes out there that you could grow as an annual. But most of us who grow radishes don't like to see that. And it's frustrating as a grower to have a bunch of bolting radishes. So selecting for biennialism, I think, is something that will improve a lot of the growing of pretty much any radish. They also, I'll say, radishes express, there's very different phenotypic expressions depending on the cultivation and soil. So even one variety that's being saved by one person grown in two different areas or two different parts of the soil or different amendments, the crop will look quite different. So it's not always because of the genetics that a crop doesn't work out or look good. The cultivation method has a lot to do with what the radish looks like. And as mentioned, the seed production can be done as a biennial, as an annual or biennial. My preference, if you can store your radishes over winter, is to grow them as a biennial. And the main reason for that is I think you end up getting a lot more seed when you have it going to flower in the early season than when you have it going to flower later in the year when it's hotter and you just don't get the quite the seed set. Last year, we ran a little experiment on the seed farm. We grew some astragalus radishes. We planted them very early, and then we noticed some were bolting and some weren't bolting. So we left the ones that were bolting to make seed. We pulled out the ones that weren't bolting and we vernalized them, and then we replanted them. So we've now saved seeds from two populations, one that was bolting early and one that had a little less bolting. And what we'll do is plant those two out and see if we can tell the difference, see if we've managed to make a selection that's moved us more towards a less bolting radish. Okay, so I'm going to get into some stories about seed breeding. And mostly I'm going to be focusing on the next one, and because it's one that I've had more experience with. But this is you can probably find this on our website or on the Seeds of Diversity website. This is a study that a breeding project that was started by Dan Brisbois from Tournesol. So he is breeding this rainbow landrace tatsoi, and I think what he had happen was that there was the original population came out of just sort of a selection he saw. There was a variety that Wild Garden Seeds released called, I think it was called Pink Petiole Mix. And really what it was a cross between a whole lot of different varieties, and it was kind of a wild landrace. It was, you know, if you planted in the field, they all matured up at a similar time, but you walk down the bed, the individuals were very different. And when you find a variety like that or a landrace like that, it presents us all with a really interesting material to be able to do our own seed saving from. So if you send out a landrace to 10 different farmers and you ask them to select for what they like over a decade, you're probably going to develop 10 new varieties. different sort of tatsois. And so that's sort of what happened here. It was a tatsoi crossed with some of the population from this pink petiole mix, and Daniel really liked this sort of purple veined and purple leaved tatsoi, and the process is mostly just been planting them year after year and trying to select the seeds that look the most uniform. You'll note there, like, and when he sells the seeds, there's a special note and it says rainbow tatsoi is still very variable and should expect a fair number of off types within your population. So the picture shown to the right shows sort of the full spectrum of what to expect. And again, as somebody who's grown a lot of different salad crops, I know that a bed like this is perfectly fine to harvest. I don't need every plant to look the same. In some ways, it's really useful to be able to go down a bed like this and harvest baskets of a salad, a variety of salad that all actually looks different, and it makes the salad look really diverse. So while you're working on these projects, especially if you are a grower and selling food too, remember you sell all of your mistakes, you sell everything that you cull, you eat what you cull, and so there's really not a lot wasted in the process. Okay, so next slide. So this is, I'm going to go through this. This has been probably my oldest teacher. This project started in 2004. I was just getting into seed saving. I was working at a farm. They had planted, the year before I got here, they'd planted a crop of Brussels sprouts. We didn't really have a market for it. We finished delivering by October, November that year. So all these Brussels sprouts grew in the field. Nobody harvested them. And I thought, what an amazing opportunity. I'm going to save seeds from those Brussels sprouts. I have the variety written down somewhere, but I couldn't find it. But the original Brussels sprout wasn't a fairly popular open pollinated Brussels sprout. So I thought, great, I can save seed from it. So I did. I saved seed. And that winter I went to a seed saving workshop, and we were talking about Brassicaceae, and somebody was talking about the possibility of producing an open pollinated white peacock kale. And we started talking about which species cross with one another. And as I sat there, I pictured the field that I saved the Brussels sprouts from, and I realized I did not have Brussels sprouts. I had something else. That field had collards flowering. It had, that was probably the year we had the most Brassicaceae flowering at the same time during that period that the Brussels sprout flowers were maturing. And so we had crosses of all kinds of things, of lacinato kales and collards and this white peacock kale, and there was a purple peacock kale. So I realized I did not have Brussels sprouts. I had something else. And we used to grow a lot of salad crops, and I didn't like the idea of growing hybrids, but for producing a really pretty salad in the spring, these white peacock kale are very beneficial. They make it through the winter. They're incredibly hardy, and you get all this like beautiful white leaved sort of sprouts, denticulated leaves in the spring. So I was kind of interested in taking on maybe the goal of trying to breed a white peacock kale. At the workshop, we were told that it's nearly impossible to do, which is probably why I decided to try. And so that's what I started to do. So the first year, I took those Brussels sprout seeds, I planted 20 flats of them. There's 100 plants per flat. So that was 2,000 plants. And I selected down to 100 plants. So I got rid of 90% of, or more than 90% of what I grew. And what I was looking for, just at a very small sort of stage as they were growing, they probably had 4 or 5 true leaves on them. I was looking for leaf shape that looked similar to the white peacock kale. It's funny how over the years, the more you observe plants, the more you learn about what to look for. And now I know that when I look at those plants in the very early cotyledon stages even, I can tell what color they're going to be sometimes. based on the kind of tonal color of the stem, because purple leaved plants will have a purple stem. And so you can often tell those characteristics earlier on. And I think that just comes from lots of observation and the help of a few good friends who suggested I should look for that. So the picture here, the top picture there of the white peacock kale hybrid. So when you plant this out in a field, every single plant looks almost exactly like that. They mature up almost exactly at the same time. The other pictures in here are probably about 2 or 3 years after selection from that Brussels sprout. So you can see there's still tons of diversity. There's some wide-leaved plants that I was trying to select away from. There's some purple individuals, which because I was trying for a white peacock kale, I was selecting away from purple. In hindsight, I kind of wish I had segregated the populations and been working on a purple version of this, because we really like that color in it. And even in what we produce still, there's still that purple does come through. So this is just saying, this is how many years, 18 years or so after beginning this experiment, and it's still a wild population and not a very uniform variety. And so what I've been doing over the years, trying to figure out how to make it more uniform without having something that's inbred. And the goals of this project, I didn't know exactly what I was wanting to like what even I was trying to develop when we first did this. At first, it was just a white peacock kale. And then if you click to the next slide, this is sort of later in the season. This is what ideally the plants look like. These were obviously selected out from a fairly large population. They didn't all look like this. They didn't all produce sprouts like this, but quite a few did. And so I was selecting for this. And then one winter, we had such a cold winter. I can't remember how many years ago it was, maybe 12 years ago or so. I've rarely, in our environment, I live in the lower mainland, kale usually makes it through the winter. It does get damaged, and maybe 50% of the plants make it, but 9 out of 10 years, 50% of the plants at least make it. Sometimes they all make it. This one winter, every single kale in the field died. I think I went through the whole field because I was also trying to save for lacinato and managed to find 50 lacinato plants out of probably a 1/4 acre of kale. However, not a single one of these died. They all made it through the winter. And so it made me become aware that what I'm also selecting for in terms of a useful plant is something that does actually make it through our winter very reliably. So it was just neat to see that. So there's a question, how big is my growing space and how many experiments are going on at a time? Probably too many. And since then, we've been able to move some of the experiments to the research and education seed farm run by Farm Folk City Folk. These kinds of projects are really hard, I will say, for a farmer to take on. As a farmer, I never got paid for doing any of this. I'm not patenting these varieties when they come out, and I'm not recovering a lot of costs. So it takes a lot of time to do these experiments, but like I say, I don't think there's a better teacher out there. So I see this as paying for a really good course. I grow on the farm I'm on is 20 acres. We grow on about 5 acres, but there's probably 1/8 of an acre is what at any given time that we've grown seed crops, and a much smaller amount of space for these types of experiments. But I do have the ability to have, say, a 200-foot bed 3 feet wide to do an experiment like this, and I can fit 400 plants in. So it does give me the ability to do that here. I'll say if you're doing something like this in your garden, though, when you cross 2 very different species together, especially during the early stages, you don't have to worry about having a very huge population because you've got such a huge mix of genetics that came from those 2 different varieties together. So you're not going to get a really inbred plant, even if you just save from a few. As you get out to the stage where they're more uniform and you're able to maybe call it a variety and you want to maintain certain characteristics, that's when you want to move to like a much larger population for selection. So that is what we were trying to do. So I was trying to make these more uniform, and the method that I've tried on this twice over the last 18 years. So most of the time I just grow out a bunch of these in the field. I select my favorite, say 70 to 80 plants, and I dig them out in the spring and I move them to another field so they flower without crossing with the ones I didn't like. And that's how I've been saving seed. But 2 seasons, I believe 2 seasons, we have tried this technique called progeny selection. So you can just skip to the next slide. So this is a way to work on creating more uniformity in a variety like this that's outcrossing without inbreeding too much. So following this, in year 1, all of these X's are individual plants in a field. And I highlight, say, my best plants here. I've just selected 5, but I'm probably going to save a few more than that. Those plants I let go to seed. Now, if I have the space, I could dig those out of the field, move them somewhere else so they don't cross with all the rest. Depending on what the crop, that might not be possible. So if you're doing this type of thing, say with that Tatsoi variety that Dan Brisbaugh was working with, probably they're going to all be flowering in the same field together. You're going to be observing them too close to flowering time to be able to dig them out and move them. So they actually will all cross with one another. However, that still means that when you find a plant that you really like, you at least know that half of the genetics of that plant are going to be good. You don't know where the pollen came from, but you know that the plant that produced the seed looks good. So you've got at least half the genetics that you want. So what we've done is we've saved seeds from these individuals and we bag them separately. So we don't mix them together. We label them separately. So red plant gets labeled red plant and green plant, green plant. And then we plant rows of these out the following year. When you plant the row out, what this will show is uniformity within that family. So if I plant a row out and, in the red row, maybe there was one individual that looked amazing, but there was a lot that didn't look like what I'm hoping for. So in my what I was aiming for with this kale sprout was white, very denticulated leaves. In some years, I was selecting for sprout formation, but for the most part, I've been selecting just for leaf pattern. So I've been selecting away from some of the wider shaped collared shaped leaves. And so if I got in this red population, if I got a couple really great denticulated white leaf plants, but there was a lot of uniformity and a lot of the others had wider leaves, I know that that's within that family. And so I will not plant any of those out. I won't even take the really good ones because I know that those also have the genetics of the rest of what I've observed in the family. So I will get rid of that entire family of plants. And then of the other ones, if they're uniform enough and producing individuals that I really like, then I'll dig out the nicest individuals from that family and again, move them to a field somewhere so that they can all cross together. And then bulk the seed up and plant it out and see where we've gotten. And hopefully it's more uniform. Now, if you weren't able to separate those plants out, so in the year 1 plant, if that red plant, say, was crossing with the green plant, or all the other individuals were crossing with them, and you weren't able to dig them out and isolate them the next year for seed saving, it means you've also got all this other background genetics in that you want to minimize. And so what you can do is you take your year 3 plants, you bulk them together, and you plant them again in the field, but you've kept all of the plants from year 1, labeled individually. And so you know that you really liked the parent green and the yellow and the blue and the beige one or the orange one. You've gotten rid of the red one, but you've kept those seeds from 2 years ago. And then you plant those seeds out with the seeds you've bulked from year 3. And this is called a back cross. And what that does is because you know that half of the genetics from those plants in year one was good, you're kind of going back to that and you're putting more of those good genetics back into the variety that you've had. Again, there's always going to be background stuff, and this is something you do probably a couple times year after year. You get them a bit more uniform, and then you should be able to move on to more of a population type breeding. So if you go on to the next page, I'm just going to show the process of this. So this is us digging plants out of the field. These are the individuals that we liked. This is an isolation tent at our research and education seed farm. So this excludes insects, which means that they won't be crossed with the other Brassicaceae species that we had flowering in the field at the same time. However, keep in mind, these things are insect pollinated, so you can't have no insects in your cage, and that's what creates the huge amount of work in order to not have these things crossing, is you actually have to introduce insects into the cage. So you can buy flies, is often what people do in labs or in breeding projects, or sometimes for greenhouse pollination. Or we attempted, and it is possible to find some roadkill and create your own flies. As gross as it is, it's certainly possible. It was trickier than I thought, though, because the flies actually they need a warm environment to produce. So we're trying to produce flies early in the season, and most of us don't want to have these things, these types of experiments happening inside our house or under our bed. And so, yeah, it was a little tricky to try to produce flies for this. But this was the process anyway of doing that. And yeah, so this is what happened. Oh, so there's a question here. How far apart do you need to isolate families? So the Brassicaceae, like they're insect pollinated. So I don't usually like to, it's hard to give an exact number, like space distance, because depending on your topography, it might be closer or further. If you're in a big open field and you have tons of bees around, you're going to need more space than if your farm has small pockets of fields separated by forest, but they will cross quite freely. Our farm is 3 acres, and there's no way we could grow 2 Brassica species there without there being a good chance that they're going to cross together. So again, you can use things like insect exclusion. The other way you can do this, if you have 2 Brassicas growing in the same field at the same time and you want to save seeds, you can rotate which ones you open and which ones you close. So if you're able to cage them with some insect barrier, and say I'm growing a lacinato kale and a cabbage seed in the same field in the same year, I can cover them both, and then I can keep one open for 2 days, close it up for a day, open the other one for 2 days, close it up for a day. So you rotate which ones are exposed to the insects, as long as you give them a bit of overlap time so the insects aren't able to carry pollen from one plant to another. It's not easy to exclude insects. They're small, they can get in, but any barrier mitigates that risk of crossing. This isn't a great picture, but what you're seeing here are 2 different populations of these plants. So when I produced these seeds early on, I shared out this breeding project. So I gave seeds away to a lot of different people. I think it was in 2007. So on the right-hand side, this is this was grown out from seeds that we got from Sunshine Farm in Kelowna. They got this seed from me in 2007 and have been doing their own selection. I don't believe they've done any progeny selection. And so they've kept it a bit more diverse, and there's some more wide shaped leaves in this. And so they got it in 2007. I think they saved it maybe 2 or 3 times. This seed was last saved in 2014 that we planted out last year. The germination was still really good on it. But it was interesting for me to be able to see that we have actually made some progress. So on the next slide, there's a bit more close-ups of it. You'll see what they look like. So On the left-hand side, this is the population from our seed farm. You can see so these are photographs of like typical individuals in the row. You can see there is some purple in it, but most of them are pretty denticulated, kind of furry-leaved individuals. And the population on the right from Sunshine Farm, there's still a lot more of the wider leaves. You do see some of the individuals quite similar to the farm folk population, but there's also just a much wider variety of plants that are have those original characteristics that we've been trying to breed away from. So I think that takes me to the end of this presentation. And yeah, these have been sort of my teachers, and I encourage you to all find some of your own by delving into this and making crosses and playing with these species. So maybe we can stop screen share. Yeah, great. And we can do some questions if people have any questions or comments. I'm really curious, especially to hear if anybody else has done any breeding experiments or projects that they'd want to share about. I'd love to hear that. Or if there's any questions about any of those families or species, we can talk about those. How do you ID inbreeding so you don't save from them? I might not understand that question. Inbreeding, I guess, is something that happens when you save from too narrow a genetic population for too long. And again, depending on the type of plant, some plants like, lettuce and tomatoes, you can have a very small population, and some of these you need a larger population. You can identify, I guess you identify an inbred population by a major lack in vigor. The time that I've seen this is saving from too few carrot seeds. I think I saved from a population of 7 twice in a row, and the vigor on those seeds was pathetic. They just didn't grow very well. So yeah, you'll see them in the growth stage, I guess. I don't know if that answered the question, but feel free to clarify if it didn't. And if people want to unmute themselves and ask a question without typing it in, you're welcome to too. Yeah, so just to clarify, I'm saying rip out the whole plant. So the plant with as much, root and soil on the root, and I would put it into, I usually put them into big totes and then heel the soil in so that the roots in, so that you're keeping the roots nice and moist over winter and keep it living.
Yeah. You take off the leaves too, so you don't transpire. Otherwise, you'll lose a lot of moisture through the leaves. Right.
Yeah. The screens used for threshing. Yeah. So again, like I say, brassica seeds are quite easy to separate. They're nice, round, very round, heavy seeds compared to flat, light chaff. So when we use screens for like really precision seed cleaning, it's important to get really precise sized seeds. For cleaning brassica seeds, you could go to the hardware store, get hardware cloth in different sizes. Just something that's small enough to let the brassica seeds through and most of the chaff not come through. But again, once it's been threshed, it's very easy to separate through a winnowing process too.
Yeah, the winnowing can be done with a variety of things. Even a air compressor in a bucket works really well for brassicas because the pods are so light for so many.
Yeah, right, that's true. So that method Lisa just described, you put your seeds and chaff in like a Rubbermaid tote or a bucket, and if you have an air compressor, you can spray in the bucket. The chaff will fly out before the seeds will. It's an easy way to clean seeds. Somebody asked if we could just put the first slide up again to remind us of the different species. So I think the slide they're referring to is slide 6, maybe. Yeah. So yeah, these are,
Daisy is coming to see me.
Yeah. Daisy, the
I
pose.
love my Daisy. Goodbye, Daisy.
somebody's asking if we run tours at the seed farm. I'd love to see what you have there and how it all works. We do run tours at the seed farm. We're considering scaling back at the seed farm a bit, or actually diversifying out. So we're hoping to have more than one location where we'll do some of this work. And by even next year, this is coming from the Gulf Islands Food Co-op, there might be a location closer to you that you can that we can meet in and see some experiments and seed saving experiments. But for sure, if you're interested in coming out to the seed farm in Abbotsford, you could do that too. We do have some open house days that we do there, but if you're a group wanting to have a tour and you're interested in seeing this, please just get in touch with us and we can arrange to do that somewhere. Okay, so there's a question here from Elena about, this is a relatively new to seed saving. She's running a project for a nonprofit. Her sense is that they're too new at this to save anything but selfers, and that they don't have enough space or winter storage. But they want to understand the process. Is there a small scale type version of these experiments we could try? Absolutely. Again, the red Russian kale, you can save from quite a few plants. Crops like within this Brassicaceae species, arugula is a really easy one to do on a small scale. You can plant a lot of individuals in a small space. You can even harvest the greens and eat them and then let them go to seed later in the year. They will flower all in the same year. They're an annual. They're a great, nice, easy crop to save seed from. They're very generous in the amount of seeds they produce. They don't, again, it's easy to plant lots of those. So that's one that I'd suggest if you're in a small situation like that. It might be really good. And I don't know, I think that even some of those, I think even some of the, something like broccoli, you might even be able to try. Again, you won't be able to harvest the broccoli, but you can identify a couple individuals to let go through the season and save seeds from them. Somebody's got their hand up. Julie, do you want to unmute yourself and you can?
I do, thanks. Thank you so much for this session. It's so very interesting. I'm actually coming to you from Calgary. I'm part of a seed saving library, and we were just talking about radish seeds actually. So it's really hard for us to do biennial seeds here. I've done it a couple times in my greenhouse, but it's a bit of a process to get them to stay over winter. We're also in a really unique area where I'm located, and the seed library actually is at the foot of Chinook Arches. So we're right at where the Elbow and the Bow Rivers meet. So we have these weird fluctuations of temperatures. So for example, 2 weeks ago, it was -25, quite literally for 4 days. And then today it's like +3. So I'm gonna go peek in my carrots. I have them in a like a cold frame, and I planted them late. Well, we had a really freaky season. It was super hot until the end of October. So I planted them probably at the beginning of August, thinking that I would harvest them, but now I think I'm going to try and keep them throughout the year in the cold frame and see if I can keep them again for next season to see if they can do seeds. So I'm just curious about, like, I didn't plant them. I don't know how to do things in straight lines, but I planted them kind of in an area. And so if I can get those to go to seed, I'm just wondering, what do you think would be like, I'd say I have about 100 carrots. If I was going to harvest those seeds next year, how many seeds do you think I need? Like, how many plants do you think I need to actually find out whether or not they're A, going to germinate, and 2, are they going to grow carrots?
Yeah, so I mean, for carrots, you're looking at a population of about if you could plant all the 100, that's probably good. That is around the population size that you'd want. And
Okay.
for carrots, you want to space them out quite far for planting for seed. So even, well, you could put them a foot apart, but they could use
Okay.
even more space than that. So you're looking at spreading 100 carrots out quite far for seed production. I will mention, like, you can harvest the carrots and keep them in a cooler over winter. You just have to make sure that when you prepare them, you leave a growing tip on the top of them. So again, as this would relate to brassicas, you know, you want to take away as much of the green growing part on the plant, like the cabbage leaves, but you want to be able to make sure you maintain a growing tip. I don't know if that answered your question, but
I think so. So when you say growing tip, could I take like the top of the carrot, like we probably eat the bottom of the carrot, but the top of it where the leaves aren't, and just have enough so that it stays alive,
yeah.
and then use
Yeah,
that, and then
So just like, yeah, even if there's like a centimeter of green at the top, you
Okay.
just don't want to cut that little growth tip. So, you know, you
Okay.
can peel the bigger leaves away and then keep the center one.
Okay.
Yeah. Yeah, and you
Great.
can store that in a cooler, you know, if you didn't have space to, if you're challenged to keep them outside. I know, yeah, you could potentially keep them in a cooler. But you
Like a
probably want to sort of heal them into something so that the tip is sticking out, so that the
okay,
tips don't rot.
like a cooler. Do you mean like a? Excuse my, I don't know what that means. Like
If
a.
you, well, I guess something, if you had a walk-in cooler, if you have a fridge, you know, a fridge size thing obviously
Oh,
isn't quite big enough, but if you have a walk-in cooler, you could put it in there, or again, a root cellar, anywhere that's going to stay around 4 degrees and not freeze. Carrots can even handle it colder than that, but they can't freeze hard over
okay.
winter. So yeah, usually what I do is I'll take something like that, put it in a bin, and with like peat or some kind of maybe sand in there, and heal them in so all the tips are sticking up and able to grow. But you can easily fit 100 carrots in a Rubbermaid tote that way, or pretty
Okay.
close to it, yeah.
Awesome. Thank you. I'm gonna try that. Thank you.
Okay, no problem. There's a question: what are the chances of interspecies crossing happening by chance? Is there a way to encourage it? I don't know the answer to that. I don't know that I've seen it happen. I have had a farmer tell me actually about that Brassica oleracea. They were convinced it crossed with a Mizuna. I was skeptical. I don't know if that's true or not, but they were adamant that it had definitely crossed with the Mizuna. So again, I think I'd have to do a bit more research, but I have a feeling if I dug into that, it's going to be very, very rare for that to happen. I think even some of the, I don't even notice wild radish plants crossing with the radishes that we're growing. So yeah. How do you know when the seeds are ready? That's a good question. The seeds are ready when they're like dry and falling out of the pods, but you don't want to wait till they're falling out of the pods, obviously, to collect the seeds. So when are your seeds ready? It really depends. My seeds were ready at one farm this year when I noticed the birds were starting to eat them. So my seeds are ready because I got to get them away from the birds. They weren't as ready as I would have liked them to be on the plant, but they were going to be, that was going to be the only way I'd save any of them. So I harvested them a little early, as long as the pods are filled out. And if you open the pod, in my experience, as long as they've started to change color, kind of like a tomato has started to become red, then you'll probably get decent seeds. So again, ideally they're dry on the plant, but they haven't started to drop their seeds yet, and you collect them then. But you really have to, it depends on the season. We had broccoli flowering this year quite late into the season. We didn't think we'd actually harvest any, but we had that ridiculous August weather in October that allowed us to harvest it. If we hadn't had October weather, we probably would have pulled a few very immature broccoli plants inside to a greenhouse, pulled out as much of the plant as possible so that the seeds can keep maturing inside. But as it turned out, we were actually able to, some of them were dried up enough to save. Isolating a crop in a greenhouse. Yeah, you can. And yes, some insects will come in. You're going to be, again, you're going to open and close the door or whatever. Greenhouses, they might not be completely sealed to insects. And you probably don't want to keep them closed all year as the plants are flowering. Often they're going to be flowering in a time of year that's really hot. What I have seen people do to use greenhouses for this very purpose is they'll roll up the sides of the greenhouse if it's a roll up side, or if you have a window, you can open the window and just put insect netting. Or in the case of this greenhouse, it was roll up sides and they covered the whole side with, I think, like a row cover insect exclusion barrier. So the wind and the air could get through, but the insects can't. Will they keep maturing? Okay, so if you cut the whole plant and bring it inside a shed to protect them from birds, will they keep maturing for a while? Or does it stop when you pull it from the ground? I'd say, again, the more plant you pull out of the ground with the seeds on it, the more energy is going to continue going into the seeds. So they will mature after you pull it out of the ground. They won't go from like flowering to seed. And if the seed pods are too immature, they probably won't finish up. But as long as they've started forming, I think that you can probably pull them in and they'll make seed. Again, it's really ideal if that can just happen in the field. Yeah, so there's a question. It seems like saving seed from just 1 or 2 biennial plants, like in a home garden, would lead to a pretty weak plant. Yeah, like in terms of inbreeding. So here's another strategy that as a community, we can work with in order to reduce inbreeding. So Julie, if you're growing seeds of one of these biennials and you have too small a population, find somebody else who's in the same boat as you and share the seeds. So you have too small a population, somebody else has too small a population. Find as many people as you can that have too small a population, bulk your seeds together and then share them out. And in doing that, you've actually increased the genetics for everybody. You're also mixing your selections together, keep in mind. So you might be selecting one way and somebody might be selecting another way. And so, it's a group project that you'd have to coordinate a little bit, but that would be a way to do a little project like that coordinated through a group of people without inbreeding your plants. The other thing I'll say is, like, if you're saving seeds for your home garden, don't worry too much about inbreeding. If what you want is just to produce, seeds for the next year, they'll still be fine. If you do it year after year, then probably you're going to be seeing some inbreeding depression. One of the ways to mitigate that is save your seeds this year, but get some seeds from somewhere else to mix into that population next year. So you're always introducing, the new seeds from that same variety from elsewhere, and that should increase. Is there, Julie, do you have your hand up again, or is that from?
I do.
Yeah.
Sorry.
No problem.
I know this is a Nebraska question. So in our neighborhood, we have a lot of people that grow hollyhock seeds, like a lot of hollyhocks. And in other areas, so we're in Calgary, I have not seen this. So we mixed them, the seeds, and now they've grown probably 5 or 6 seasons. And I'm very curious about the colors of the flowers, because I can never guarantee what flower will show up, like the color. So for example, my seeds I got from someone who grew them 4 or 5 seasons in Calgary, and hers are like red, dark purples, and some white. And when I grew mine, they were like white with pink stripes. Some were light purple, some were like a light pink. So I was just curious about, once we have mixed them and we sort of circulate them and we collect them again, Do you know anything about why they would be different colors and how how that all works?
I only know that colors in plants is very complicated, and I don't know the genetics of how hollyhocks cross, but color is often composed of multiple genes, so you'll get like a variation in colors. It's not like there's one gene that's pink and one gene that's purple. You get gradients in between from different plants. I'm thinking more like chards and stuff like that. When you cross them together, it's not so cut and dry like red chard, gold chard. So I don't know what the characteristics are of hollyhock. If they are outcrossing all the time, I would expect you to, if you're saving seeds and bulking them each year, you're going to probably move to a more uniform color. And if you want to maintain a specific color, you need to, this is where the work of like observation and what I'd argue is like seed breeding work comes in. Even if you're trying to do a good job of saving it, if you're trying to grow a black hollyhock, you need to make sure that you're saving seeds from just the black hollyhocks and getting rid of the other colored ones. So if they cross, again, I don't know, you'd have to look that up. It's a little different for each plant.
Yeah, no, it's all good. I can say that they're quite drought resistant now, the ones that we've had for some time. And they're also really hardy because we have the weirdest weather here now. So I'm also curious about the radish seed. You mentioned like you would actually take them out of the ground and replant them again. So is there a particular type that do you think would be useful, or not useful, but would make it through? Like we're in zone, I think 4. I'm just curious because we have a lot of people that have donated radish seed to our Calgary seed library, but I'm just not sure about what kind of radish it might be, and maybe we want to bulk up that particular seed.
Right. Is that like when they submit radish, do they submit it with the variety name, or is it just you've got a whole lot of seed that says radish?
I'm always so hopeful that they give us more than just radish.
Yeah.
And some come with the pods, which is fine. But yeah, we don't know what they are. But for my own radish seed saving, I think I've used the white one. It's like a variety of daikon, I think, but that it takes a very, very long time.
Yeah. So those ones, a lot of the daikon, like same with the black radish and the watermelon radish, those are slow growing, but they store really well. So plant a late season crop of those, and kind of like the carrots, dig them out of the ground and store them in a cooler or some cool space. Same idea. Take away as much of the greens and leave as much. Just leave a growing tip, and you should be able to get them over the winter and replant them. Some of the faster radishes, like your typical red or French breakfast or whatever, those are ones you could get away with probably planting really early in the season and letting them flower all in one year. But yeah, they differ a fair amount based on the different variety of radishes, their flowering time, their bolting time, and their biennialism.
Awesome. Thank you so much.
No problem. There's a question here. What time of the growing season do you replant your pulled plants? I guess it depends what you're replanting. Let's see, cabbage and that. I mean, we've got them actually in the ground outside right now. I guess it depends on where you are. They can handle a little bit of freezing for sure, but probably not a really, really cold, long freezing. And they don't like a lot of wet and then freezing, something like cabbage and that. So I think in the spring, I'm probably planting them out where I am. I'm planting them out in February or March. But whenever your chance of freezing is over and you can get them into a field. And I guess it depends on which varieties too. So that would be for like hardy Oleracea. Although I imagine like, we're planting, again, out here, we're planting radish probably in March, even outside in February in greenhouses. So that timing should work too. In the interior, I haven't grown a lot in the interior, so I'm gonna guess maybe in April or something like that. Brussels sprouts. Okay, here's a good question. I've saved Brussels sprout seed this year. Will they have crossed with wild mustard? I don't believe so. I don't think wild mustard is in the Oleracea species. And again, I haven't seen a lot of those really wild species cross in. Even the wild radish, I don't see cross in with our radish. So I think you're safe. As long as you don't have any collards or lacinato or those kinds of things flowering, you should be fine. Will wild mustard cross with anything? I don't know. Is there anybody else? I mean, I'm sure it'll cross with something. I haven't noticed it crossing in. Like I haven't seen. I've saved tatsoi seed, mizuna seed, saved mustard seed. I know we have lots of wild mustards growing on the farm. I haven't seen that type of cross contamination happen. So, I mean, in theory, you would think that they would if they're the same species, but like I say, I haven't seen it happen. So
I don't think they are the same species.
the wild mustard and the wild mustard, is that more a nigra, like a black mustard relative?
I think it might be in a totally, I'm not sure, because most mustards are like the Brassica junkia or Brassica nigra, but some of the wild mustards are like a totally different genus, like the
Okay.
Sinapis arvensis. But I'm sure there's other wild mustards too.
Have you ever noticed that, Jolene? Have you noticed crossing or?
No.
We're not hearing your sound, or at least I'm not hearing your sound. No, okay. Yeah, then I'm guessing you're probably safe then. Oh, okay. Dijon, that would be interesting to see. I'm curious because that is a different that's in the black mustard. Is that in the black mustard or is that, do you know what type of species Dijon is? Do you sell that through? Yeah, I think it's Nigra too. So there's probably not a lot out there. That's cool that you're growing that. You're selling that through BC EcoSeed Co-op? Oh, nice.
Great. Well, with 5 minutes left, I'll just let everybody know that our next workshop is Saving Biennial Seeds, and it starts at 11. Also, just wanted to mention that our Citizen Seed Trial registration is up. I'll plop that in the chat. So we're looking at radish, peas, and salad turnips. And I'm also just going to send out a survey link if you wouldn't mind evaluating how you found this session. And then it looks like we still have another question. So
Yeah, so can I replant cabbage for seed and grow rutabagas in the same garden? You can do that. If you're growing both for seed, then they're going to cross with one another. If you're just growing the rutabagas for rutabagas, then, oh, actually, never mind. I think I'm wrong. Those are different species, aren't they? You could actually grow both those seeds together and save them. So I think cabbage is Oleracea and rutabagas are Rapa, or yeah. So yeah, that would be fine. And again, even if they were in the same species, say you were growing cabbage and kohlrabi, those two things would cross. If you're growing the cabbage for seed and the kohlrabi is in its first year, obviously there's not going to be crossing happening. If they're flowering at the same time, they'll cross. But Okay, great. And yeah, it's super helpful if you guys can fill out the survey. It really helps us know if we're on track or if there's anything else we can do to make these sessions better, help out. Rutabaga and red Russian kale can cross, right? All right, well, thanks, everybody. And again, I encourage you all to just go out and play with these teachers, because you'll learn a lot by making lots of mistakes over the years. And yeah, every year is a new learning experience. So. Okay, and hopefully we'll see you in, is it half an hour for a session on saving biennial seed.
Thanks for coming.
Take care.
Thank you.